Welcome, everyone. Thank you for joining us for today's webinar, The Phase 2 Endpoint Dilemma, Bridging Subjective Scales and Objective Measures in Neurodegenerative Trials. I'm Heather Masaccio, your host from Clinical Leader. I have just a bit of housekeeping before we get started. If you have a question for our speakers or wish to share any thoughts of your own, please submit it through the Q&A box on your screen. Our speakers will address those questions at the end of their presentation. If you run into any problems with the stream today, please try refreshing your browser first. If that problem persists, go ahead and use that Q&A tool to submit a question, and I'll help get you back on track. This event will be available on demand. You'll receive a link via email as soon as it's available. Now on to our speakers. Today we are joined by Francesco Bibbiani, MD, Vice President and Therapeutic Area Head, CNS, and Marty Kraut, Ph.D., Director of Operations CNS, both with Center of Excellence Avance. I am going to turn it over to Dr. Bibbiani to begin the presentation.
Great, thank you. And good morning, everyone. And thank you for joining today's webinar. My name is Francesco Bibbiani. I'm the Therapeutic Area Head of the Advanced Clinical CNS Center of Excellence, and together with Marty Kraut, Director of Operations, we're going to tackle one of the most challenging aspects of neurodegenerative drug development, the endpoint selection in phase 2 trials. Let's start with the reality check. Neurodegenerative trials face a perfect storm of endpoint challenges that don't exist, or at least not to this degree, in other therapeutic areas. First, slow disease trajectories. Alzheimer's, Parkinson's, ALS, these diseases progress over years, not months. Second, limited trial duration. Your phase 2 trial typically runs 12 to 18 months. And you're trying to capture a signal in a narrow time window that might not align with the natural course of the disease. Third, high patient heterogeneity. Two patients with the same diagnosis can progress at completely different rates. One might decline rapidly, one might remain stable for years. This heterogeneity dilutes your treatment signal and inflates your sample size requirements. Finally, regulatory acceptance. The FDA and EMA have clear preferences for validated clinical outcomes. Novel endpoints require extensive justification and validation, which takes time and resources that most biotech companies don't have in their phase 2. These challenges translate into sobering statistics. According to BIO's 2021 Clinical Development Success Rate Analysis, the overall phase 1 through approval success rate across all therapeutic areas is approximately 8%. But for CNS, it drops to 5.9%, the lowest of any therapeutic area. And the biggest drop-off happens between phase 2 and phase 3. Where only about 29% of programs, they actually advance. Let's talk about why the scales we have relied upon for decades are increasingly problematic in modern phase 2 trials. Insufficient sensitivity in early disease stage. Most legacy scales were developed and validated in moderate to severe populations. When you apply them to prodromal, or early stage patients, they simply don't move enough over 12 to 18 months to detect a treatment effect. The ADAS Cog was developed for moderate Alzheimer's dementia in the 1980s. We are now testing drugs in amyloid positive individuals who are cognitively normal or have MCI. These scales were not designed for this, and it shows. Floor and ceiling effects. If your scale can capture the range of impairment in your population, either because patients are too mildly affected or too severely affected, you lose statistical power. It's like trying to measure millimeters with a yardstick. High inter-rater variability. Subjective clinical scales depend on rater judgment. Different raters interpret items differently. Even the same rater can be inconsistent across time points. This variability directly erodes your ability to detect true drug effects. Practice effect. This is particularly problematic in cognitive assessments. Patients get better at the test over repeated administrations, which masks underlying disease progression. Your drug might be slow in decline, but the learning effect obscures it. Then capture global change poorly over short time frames. These scales were designed to track progression over years, not months. They are insensitive to the subtle changes that occur in 12 to 18 months in phase 2 trials, especially in these slow progressing populations. Then misalignment with drug target. If your drug targets motor symptoms in Parkinson's disease, but your primary endpoints includes non-motor items that won't respond, you're diluting your signal. Global scales aren't always appropriate when you have a very targeted mechanism. Then subjective assessments can be vulnerable to bias and placebo. Rater expectations, patient expectations, caregiver expectations, all of these can inflate the placebo response rates in subjective scales, making it harder to demonstrate a drug-placebo difference. Let's make a concrete example using the most widely used scale in Alzheimer's disease trials, the Alzheimer's Disease Assessment Scale Cognitive Subscale, or as it is known, the ADAS Cog. This scale was developed by my Boston colleagues in 1983 for patients with moderate Alzheimer's dementia. It has been the gold standard for decades. But research by Podorna and colleagues published in Alzheimer's Disease, Alzheimer's Research and Therapy demonstrated 3 clinical limitations when used in MCI and early ED populations. First, poor sensitivity. The ADAS Cog showed limited responsiveness in MCI population over 18 months. This natural decline was so small that detecting a treatment effect require massive sample sizes. Second, strong learning effects. Patient improved on test over the first 6 to 12 months due to practice, masking any actual cognitive decline. This means your placebo arm often looks like it's improving, not declining. And third, the global score obscures domain-specific improvements. If your drug improves memory but not language, that effect gets averaged out across 11 or 13 items, depending on the ADAS Cog you're using, reducing your signal. The bottom line is the ADAS Cog is still used because it's regulatory validated, but it's often not fit for purpose in early stage phase 2 trials. Now let's look at the Movement Disorder Society Unified Parkinson's Disease Rating Scale, or as we all call it, the UPDRS. That is the standard in Parkinson's trial. This is actually a well-designed scale with very good psychometric properties, but it has 3 operational challenges that make a phase 2 trial difficult. First, a high placebo response in the Part 3 motor examination, which is usually the most commonly used. Research by Goetz and colleagues published in Movement Disorder showed that placebo-associated improvements of 20 to 30% in the motor function. This is driven by patient expectancies, rater bias, and regression to the mean. When your placebo arm improves by 20%, your drug needs to show at least a 30 to 35 improvement just to be statistically significant. Then second, day-to-day fluctuations. Parkinson's symptoms fluctuate significantly based on the medication timing, stress, sleep, and many other factors. This intrapatient variability increases the noise in your data and requires larger sample sizes. Then third, medication timing confounds. You're assessing patients on their on state or their off state. How long since their last levodopa dose? These factors dramatically affect scores and are difficult to standardize across sites, especially if you have multiple sites. None of these are fatal flaws, but they make efficient phase 2 trials very challenging. Now, given these limitations with single scales, composite endpoints have become increasingly attractive. The theoretical rationale is compelling. Increased sensitivity. By combining multiple measures, let's say cognition plus function plus biomarker, you can capture the multidimensional nature of disease progression. If your drugs affect multiple domains, a composite could really detect that more efficiently than any other single measure alone. Then it can reduce the sample size requirement. This is the holy grail. Research by Hazo and colleagues in the Journal of Neurology show that optimal composite endpoints require 38% fewer subjects than single endpoints in primary progressive aphasia trials. And that is a game changer for biotech's budgets. Then better alignment with disease heterogeneity. Different patients may respond in different domains. Composite can capture responders who wouldn't be detected by a single scale. Then richer mechanistic understanding. So seeing which domains respond and which don't will inform your phase 3 design and helps you understand your drug's mechanism better. This all sounds great. So why isn't everyone using composite? So let's talk about when they help and when they hurt. So composite endpoints are most valuable in 4 specific scenarios. First, when your drug mechanisms affects multiple domains simultaneously. If you're testing a disease-modifying therapy that should improve both cognition and function, a composite that captures both makes sense. You're not artificially combining unrelated outcomes. You're measuring the true multidimensional effect. Second, when individual scales lack sufficient sensitivity alone. If the ADAS Cog has an effect size of 0.2 and the activity of daily living has an effect size also of 0.2, but together they create a composite with an effect size of 0.4, you have gained real statistical power. Third, when complementary measures reduce noise through averaging. If one measure has high day-to-day variability and another one is more stable, combining them can reduce overall variability and improve the signal-to-noise ratio. And then finally, fourth, when you have a pre-specified statistical plan with clear hierarchy. This is critical. You can't just throw multiple endpoints together and hope for the best. You need a structural analysis plan with appropriate multiplicity control, clear weighting justification, and regulatory buy-in. If these conditions are all met, then composites can be very powerful tools. But here is the other side of the coin. Composites can backfire in 4 common scenarios. One's when there's unclear clinical interpretation. Regulators and clinicians struggle to interpret composite scores that don't map to meaningful outcomes. Components measure different structures with variable weighting. How do you come up with the final composite score? If you are combining a cognitive scale that is measuring points, a functional scale that is measured in a different way, and a biomarker that it's measured, let's say, in picogram per milliliter, how do you weight them? Do you weight them equally? I mean, you need to have a very strong justification to define how you measure them and you put them together. Then masking domain-specific effects make phase 3 design more difficult. Let's say your drug improves cognition, but not function. Your drug hurts one domain and helps another. And then the composite averages them to neutral. You can miss critical safety signal. How does that really help inform your phase 3 endpoints? And then finally, regulatory skepticism without a validation evidence. The FDA and the EMA are comfortable with validated scales. Novel composites require validation, showing reliability, which you can do in phase 2, but you need to have a solid idea of what constitutes a meaningful clinical important difference. And that is, if anybody has ever tried to work on a composite, that's the most difficult part. How much of that score is clinically significant? And that's where it really becomes difficult to say this number that is clinically significant. That's really challenging. Then adding more endpoints does not automatically improve signal detection. In fact, with careful justification, composite introduce 3 major problems that can outweigh any sensitivity gains. First, statistical penalties. Every additional endpoint you add requires multiplicity adjustment. Your p-value threshold gets more stringent. You need larger effect size or more patients to maintain power. Then interpretation complexity. You'll present your phase 2 data to regulatory agencies, investors, and phase 3 investigators. If they can't understand what your composite score means, you have lost your communication battle. And then regulatory hurdles. Regulators might reject your phase 3 protocol if it relies on a composite that they don't understand and they don't trust. In practice, here it is. In practice, composites can bring important benefits, but only when the justification is rock solid and the interpretation is crystal clear. When in doubt, a single well-chosen, validated endpoint is often safer. Now, let's talk about how to thoughtfully integrate different types of measures into your endpoint strategy, not as a composite score, just how to integrate different measures among your endpoints. We'll cover functional, cognitive, and biomarker measures in the next few slides. Functional assessments capture real-world impact. That's the outcome that matters to patients and caregivers. Activities of daily living scales like the ADL scale or the instrumental ADL measures and assesses independence in everyday tasks, like, for example, managing medication, handling finances, preparing meals. These are clinically meaningful outcomes that resonate with regulators and then eventually payers. You have to plan early and integrate these assessments that will be useful after approval, right? So your goal is the approval, but eventually also reimbursement. So if the payers are not convinced, then your approval becomes very much less important. Then performance-based measures like gaze speed, time up and go, or dexterity tests, they provide objective functional data. These are less subjective than the rater assessed scale and can be measured with wearables devices for continuous monitoring. And then patient-reported outcomes like quality of life scales, they actually capture patients' own perspective. The FDA has emphasized patient centricity, and PROs are increasingly important in regulatory submissions. The key is matching your functional measure to your mechanism. If your drug targets motor function, measure motor function. If it targets cognition, ensure the functional measures are sensitive to cognitive impairment. Now, cognitive measures should be chosen based on your drug's expected mechanism and the cognitive domain most effective in your target population. For example, executive functions batteries like the trail-making test, the Stroop test, the DigiSpan, they assess attention, working memory, and cognitive flexibility. These are sensitive in frontotemporal dementia and Parkinson's disease dementia. But memory assessments like verbal pair associate, verbal reproduction, or spatial memory tests, they are critical in Alzheimer's disease. Consider by their population as primarily encoding deficits, retrieval deficits, or both. These affect the test selection. Now, computerized cognitive testing using platforms like Cogstate, the Cantab, or the NIH Toolbox, they offer several advantages. Their standardized administration reduces rater variability. Automated scoring improves reliability, and reaction time measures capture subtle changes that paper and pen tests cannot assess. So one practical tip, don't just default to the ADAS Cog because it's familiar. If your mechanism targets specific cognitive domains, then domain-specific tests will give you better signal. Biomarkers. Biomarkers are increasingly essential in phase 2 neurodegenerative trials. They provide objective evidence of drug activity independent of subjective clinical scales. Fluid biomarkers have made tremendous progress in recent years. Plasma phosphorylated tau, the p-tau 181, p-tau 217 can track Alzheimer's pathology with blood draws instead of lumbar punctures. Neurofilament light chain is a marker of axonal damage across multiple neurodegenerative diseases. And then alpha-synuclein is emerging as Parkinson's disease major biomarker. These are less invasive than anything CSF derived and more feasible for larger trials. Now, neuroimaging biomarkers, they include volumetric MRI showing brain atrophy, amyloid PET showing plaques burden, and tau PET showing tangle burden, and then fluorodeoxyglucose PET to visualize metabolic activity in the brain. These provide anatomical and functional information about drug effects on the brain. Target engagement and pharmacodynamic markers, they demonstrate that your drug is hitting its intended target and producing the expected biological effect. For example, if you're testing a BACE inhibitor potential target to the amyloid cascade like Leqembi, for example, measuring CSF amyloid beta fragments, that shows target engagement. So the strategic question is, should biomarker be secondary endpoints or co-primaries? Well, we'll come back to that. Here's a practical framework used for structuring your endpoint strategy. That said, if you're not comfortable with a composite endpoint, but that must fit all the requirements we looked at earlier. If it cannot fit all those requirements, then maybe this practical framework, looking at primary, secondary, exploratory, and so on, it's the best option. So your primary endpoint should be a single validated clinical scale that regulators accept. This is your regulatory anchor. Yes, it might be insensitive, it may be noisy, but it's validated, it's interpretable, and the FDA trusts it. So for example, the ADAS Cog for Alzheimer's, the UPDRS for Parkinson's, the ALS-FRS for ALS. Your secondary endpoint should include mechanistic biomarkers and functional measures that provide supportive evidence. These serve two purposes. First, they help you understand your drug mechanism, and then your dose response. Second, they build the case for regulatory approval by showing consistency across multiple domains. In my opinion, be conservative with your primary and innovative with your secondaries. This gives you interpretability for regulators and then sensitivity for those findings. So with your multi-domain framework, you need a clear endpoint hierarchy with pre-specified statistical analysis. The primary endpoint, this is your regulatory validated clinical outcome. Success requires meeting the pre-specified alpha level, typically p less than 0.05, with the appropriate power. And then the key secondary endpoints. These are your dose response or mechanistic endpoints, usually one or two measures that provide crucial supportive evidence. You might use hierarchical testing when you only test key secondaries if the primary is positive, preserving your alpha. Then finally, exploratory endpoints. These are your hypothesis generating measures. There's a domain-specific test, novel biomarkers, patient subgroup analysis. These don't need to be positive for a trial success, but they will inform your phase 3 strategy. What is critically important is that pre-specify this hierarchy in your protocol and in your statistical analysis plan. Don't change it post-hoc. If you claim exploratory endpoints as key secondary after seeing the data, then you lose credibility. Also, implement appropriate multiplicity control. So your statistician should be involved in this from day one. So one of the biggest challenges in phase 2 is detecting meaningful change over a relatively short time frame, typically 12 to 18 months, when the disease progression can be very slow and over years. Now, 3 strategies can improve your sensitivity. First, baseline enrichment for faster progressive. Not all patients progress at the same time. If you can identify predictors of rapid progression, for example, higher baseline tau, lower hippocampal volume, or genetic risk factors, you can enrich your trial population for faster progressors. This increases the amount of change you will see in your placebo arm, making it easier to detect a treatment difference. Biomarker certification to reduce heterogeneity. So amyloid positive versus amyloid negative, ApoE4 carriers versus non-carrier, GBA mutation carriers in Parkinson's disease. By stratifying or selecting based on biomarkers, you create a more homogeneous population with a more predictable progression. Then lastly, frequent assessments to capture a trajectory. Instead of baseline month 6, month 12, month 18, consider monthly or quarterly assessments, especially for wearables or digital endpoints. This gives you more data points to model trajectory, improves statistical power, and allows an earlier detection of divergence between treatment arms. These strategies require more sophisticated enrollment criteria and potentially longer recruitment periods, but they can dramatically reduce your required sample size. Now, natural history studies. These are one of the most underutilized resources in phase 2 trial design. These are observational studies that track disease progression in untreated patients over time. And why are they valuable? For 3 main reasons. First, they provide realistic progression rates by disease stage. So instead of guessing how much a placebo arm will decline, you can use natural history data to estimate progression with confidence. This directly informs your power calculation. Second, they identify baseline predictors of decline trajectories. Which patients decline fast? Which decline slowly? Natural history studies show correlation between baseline characteristics and subsequent progression, allowing you to design enrichment criteria. Third, they serve as benchmark for external control comparisons. In rare diseases where randomized placebo-controlled trials are challenging, FDA and AMA are increasingly accepting external controls from natural history studies. You can compare your treated patients to propensity-matched naturally history cohorts. Now, beyond traditional natural history studies, real-world evidence from registries and electronic health records is becoming increasingly useful and accepted. Registry data for patient identification and feasibility. Disease-specific patient registry help you understand where the patients are located geographically, which sites have access to them, and whether your enrollment targets are realistic. By doing this, you can really select your sites based on where your patients are located or where they get better access to those specific sites. Moreover, many registries also allow patients pre-screening and recruitment. Electronic health records for progression modeling. Observational electronic health records can model disease progression in real-world clinical practice, not under controlled trial conditions. This provides complementary evidence to the natural history studies and can identify pragmatic endpoints. External control arms to augment small phase 2 trials. The FDA's 2025 guidance on natural history studies for rare diseases explicitly endorses propensity-matched external controls under certain conditions. If your phase 2 trial is small, let's say 20 patients per arm, augmenting with external controls can improve statistical power. And post-hoc validation of endpoint sensitivity. After your trial completes, you can use real-world data to validate whether your endpoints actually predict meaningful clinical outcome in practice, in real life. But importantly, before you finalize any of these strategies, you must engage with regulators. Let me be very clear about the FDA and the EMA preferences. FDA and EMA prefer validated clinical outcomes for phase 3. They're comfortable with the ADAS Cog, the CDR-SB, the UPDRS. These have decades of validation. If your phase 3 primary is one of these scales, you're on solid grounds. Biomarkers are accepted as phase 2 co-primaries with a strong rationale. The FDA increasingly support biomarkers as co-primaries in phase 2 when there's mechanistic justification and validation data, but they typically still want a clinical scale as at least one of the co-primaries. Novel composites require pre-specified validation plans. If you are proposing a new composite endpoint, the FDA will ask, what is the validation evidence? What is the meaningful, clinically important difference? How do we interpret the score? You need answers before your trial begins. So early agency dialogue is critical for endpoint acceptance. So use a type B or a type C meeting to discuss your endpoint strategy before you lock your protocol. The FDA may have specific recommendations or requirements. Now with this, I will move to the operational considerations and pass it on to Marty Kraut, the Director of Operations of the CNS Center of Excellence.
Thank you, Francesco. Hello, everybody. Again, I'm Marty Kraut, Director of Operations for the CNS Center of Excellence at Avance. And I'm going to talk to you some about just some of the operational considerations that are important when trying to implement these endpoints that you've selected. Now, Francesco has spoken to you about the strategies for choosing appropriate endpoints and some of the pitfalls associated with them and how to avoid those. But once you've constructed your endpoint strategy, that's a big step, of course, but still only one step, because the strategy still has to be implemented successfully for the trial to be able to achieve its aims. And so what this means is taking a lot of operational steps to manage the endpoints during the trial. And the endpoints and their implementation have to be managed regularly on an ongoing basis. So you can think of this really as a continuous quality management program. So it's not just a one-time setup task where you're developing your protocol, you're choosing your endpoints, you're figuring out how they're going to be implemented, and then you're done with it. It really has to be something that is actively overseen, actively managed, and issues addressed throughout the lifetime of the study. And so doing so typically involves several steps, several parameters. And I'm going to talk about each one of these a little bit. That's going to be rater training, which of course I'm sure everyone is familiar with. Strategies for endpoint monitoring, how to mitigate placebo response, which of course is a very big challenge in clinical trials, in CNS trials. How to reduce practice effects, the importance of site selection, and then the management of patient burden. Sometimes some of the scales, the amount of scales that we include in protocols can have an impact by way of patient fatigue in terms of how they respond. So moving on then to rater training. Now, rater training, it's hard to emphasize enough how important having a good, strong rater training program is for trial success. Sometimes we hear questions like, well, is it really necessary to have a rater training program? It's expensive. It takes a lot of time to set up. And we're working with sites that are very experienced, with raters that are experienced. They've administered these scales many, many times. So really, why do they need to go through the training again here? But the reality is that no matter how many times a rater may have administered a scale, no matter how much experience, given the nature of the measures that we use, their subjectivity, variability, it's very common, very normal to see inconsistencies in the way that these scales are administered, in the way that they're scored. And that really increases the amount of potential variability that you're going to see in patient responses. So there can be a tremendous impact on your outcomes if rater training is not appropriately implemented. So programs need to be standardized. As the number of sites and raters increases, the potential for intervariability increases. So really have to make sure that raters are administering and scoring scales in the same way, both across patients within a site and across sites. Even experienced clinicians will still disagree on how to score something, how to administer something. Maybe they'll use some of the common habits from their own clinical practice. And those are things that you want to avoid. You want everyone to be administering in the same way to reduce noise. Your rater training program also should cover scale content and how to administer the scale. It's helpful when they identify common errors and how to overcome them. The errors are seen so often. In one meta-analysis that reviewed thousands of ADAS Cog administrations, it was found that almost 20% of administrations had some sort of error, either in the way it was administered, the questions that were asked, or in the scoring. Similarly, with the MMSE, we tend to see a lot of errors. Again, this is such a commonly used scale, and yet raters can still vary. For example, when asking the question on orientation to place. For example, each site will agree on what's the appropriate answer, what the name of the hospital or the department, the floor that they're on. But raters will still sometimes vary in terms of what answer they accept or when they give a prompt. For example, if a patient says hospital, they need to be prompted a little bit more. But oftentimes, or at least sometimes, raters will not do that. And that's going to have a subtle effect, but cumulatively, it could have a significant effect because it may be obscuring the fact that maybe the patient really doesn't have full orientation to where they are. So they may know they're in the hospital, but that's about all that they know. But you're not getting that lack of orientation because the question is not being probed sufficiently. It's also important to include training on behavioral factors that are associated with administration. So I'll talk about this a little bit more when discussing placebo response, but training on things like not using suggestive questioning or leading type of questions. That's important as well because, again, that can mask certain responses or certain deficiencies that a patient may show if they may not be able to answer it on their own, but if they're being led into a question, they may then be able to answer it. And that reflects sometimes just an unconscious bias on the part of the rater, and that you want to be helpful. So it's just kind of a natural tendency that comes out, but something that should be avoided. Now, in terms of requirements for rater programs and for raters themselves. It's important that programs should use assessments that look at patients' ability to accurately administer and score. So, for example, having vignettes on the ADAS Cog or the UPDRS, having raters view these, score, and compare to a gold standard. And sometimes we'll see that, again, even experienced raters may either fail or have some errors in certain items. It's not because they don't know how to administer. It's just that, again, raters bring different experiences to bear. And so they may vary a little bit from what the standard is for that particular case. You want everybody rating in the same way. You want to have raters that have specific educational and clinical experience standards. Now, it'll be up to each sponsor in working with a vendor, rater training vendor, with their CRO, to decide on what those experience standards are, those educational standards. But we do know that having the clinical experience in the particular indication is better than just general experience. And having more specifically experience with the specific scales being used is more important than just having general administration experience and similar but not the exact scale. So, the experience of the rater and the training go hand in hand. But there's a lot of research to show that the training itself contributes more to proper administration and to successful scoring than the actual experience that the rater has. And again, just like with talking about managing the endpoints overall, it being a continuous process, rater training should be a continuous process as well. It shouldn't be just a question of, okay, we've got getting raters ready to go before site initiation, and then we stop and just kind of pick up again if there happens to be a new rater at a site. There really should be, to make the most out of these programs, ongoing quality monitoring, tests of inter-rater reliability, and refresher training throughout the study. So especially for studies that are of longer duration, if there's no refresher, no attempts to kind of bring the raters back to the standard, we often see kind of a rater drift within and across raters. Or if there's significant protocol amendments, that can be a reason for having further training. So again, it's an ongoing process throughout the life of the study or throughout the patient participation in the study. Now, when it comes to placebo response, again, I know we're all familiar with placebo response and how much of a factor it plays in the outcome of trials. I mean, placebo response rates have just continued to rise. Seen estimates where for CNS trials that fail, as much as 30 to 50% of the failures, at least partially attributable to the placebo response. So there are a lot of steps that can be taken to try and mitigate this. None of them are perfect, but the more focus there is on this, usually the better the outcome. So blinding integrity. I mean, raters really shouldn't be exposed to information that can reveal treatment assignment. There needs to be a limit to their being able to access other information in the patient's charts, for example, their side effects or that they've experienced, anything like that. You really just want to kind of, as much as possible, isolate them and not allow access to other information. And that includes kind of limiting the interactions between raters and other staff. Now, again, it says when feasible or as much as feasible, because we know we can't just put the raters in a box. They're oftentimes site staff. They're going to be interacting with others there. And so it's not possible to do this to 100%. But as much as possible, it is important to try and limit the interactions. Similarly, it's important to train raters and really all staff to avoid the therapeutic relationship as much as possible. This is a really big contributor to the placebo response. Again, and it comes out of just natural human behavior, natural caring, wanting to help. So sometimes, when you're interacting with the patient, and particularly if it's a patient from one's own practice that there may be subtle cues that are given, suggestive of help. The patient's going to get better. That just kind of this. Sometimes it's just the warmth of the relationship itself that can help a person feel better. For example, in depression trials, that's commonly seen. Just the idea that you're there, you're talking to someone, you're getting this kind of positive interaction from staff. Can really contribute to the placebo response. Can have patients in the placebo group seem like they're doing better, and it obscures legitimate differences, legitimate treatment effect on the part of the treatment drug. So it's really important to focus on the difference between the therapeutic relationship and the research relationship. Now, we don't want to, of course, be robotic about it. We won't want to be, creating an atmosphere where patients are uncomfortable. So it's important at the start and throughout the trial to remind patients that, this is the reason that we're doing it. This is the reason that maybe your interactions today when you're coming for the trial seem a little bit different than when you're coming for your usual office visit. Also helpful to remind patients that there really are no right or wrong answers when they're responding to an interview or even to a questionnaire. All information is valuable. It helps. Whatever they're responding, whatever they're feeling, as long as it's honest. Even if they're not getting better, that's obviously useful information. A lot of patients, of course, they're there. They want to get better, and they also want to please sometimes. Again, the natural human behavior to say, maybe I'm doing better than I actually am. So having these kinds of interactions with patients at the beginning and throughout the trial is helpful in trying to reduce that. Francesco mentioned practice effects, and of course that is something that is important, and again can contribute to the noise in a trial, because the more often, at least with certain measures, the more often a patient is exposed to it. And this is particularly true, of course, with cognitive measures. The more often they see the same items, the better they're going to get at it. So there are certain ways to mitigate this. One is using a single placebo-blind run-in. There are also scales that have parallel forms or alternate versions. And digital assessments that are becoming more common, assessments, for example, in apps on your smartphone, massing those can help reduce practice effects up front. Okay. Now, site selection, of course, is very important as well in making sure that your endpoint strategy is successfully implemented. Again, I mentioned before, it's important to choose sites that have experience with the indication, because they're going to be the ones that typically have access to the right patient networks. They'll have their understanding and familiarity of the protocol from the prior work that they've done. They'll be able to better use their clinical judgment when it comes to assessing the patients. Again, I mentioned the site rating experience with the specific scales. Again, research has shown that probably the single biggest factor is in rating success is experience with the specific scale, not just rating experience in general, and not just clinical experience with the scale. So, if you used UPDRS in practice, that's great, but having the experience using it in a research study is going to be more predictive of success rating in the study. Biomarker experience and capabilities, again, Francesco mentioned, and it's really important. Sites need to have access to the appropriate equipment. They need to have experience running these tests. They need to have experience, for example, with lumbar puncture and using the MRI machine. Most, of course, sites that we've chosen will have that, but it's important to to still assess that. I mentioned having the patient databases, and databases particularly matched to certain enrichment criteria. For example, patients known to be positive for certain biomarkers, APOE4, for example. So patients that, or sites that have patients that match those criteria, and again, sites that have done these studies in the past and have done them well and have had the highest enrollment are more likely to be the ones that do better in your current study. Now, I mentioned the implementation of the endpoint strategy involves kind of a continuous quality management. And one way that's becoming increasingly more common doing that is through centralized ratings. So in this situation, instead of having raters at each site complete the ratings, a smaller group of expert clinicians, centralized, maybe working for the vendor, are the ones that actually interview the patients, that conduct the assessment and rate the patients. And this can be very helpful in a lot of ways. Again, it'll reduce inter-rater variability because there's only going to be a small number of raters. Reduces or likely to reduce placebo response because the rater is not going to have a whole lot of interaction or any really with the patient outside of those ratings or with other staff. It's easier to ensure that the experience of the raters because again, you're working with people that are selected specifically for that experience, that work for this vendor, that have the history, as opposed to the variability you might see at sites. And sometimes, in particular in complex indications, sometimes complex or lesser known scales, it may be hard to find experienced raters. And so these centralized rating programs can help with that. And they help reduce site burden as well when it comes to things like scheduling, making sure that raters are available. We always say that we want to have the same rater rate the same patient as much as possible, but things get in the way as sometimes a rater can't make it, they leave. So having the centralized raters is important to help in that way too. But again, when it comes to quality, it's reducing that inter-rater variability, reducing that placebo response. And then ongoing monitoring during the course of the trial is also quite critical. Central monitoring, central surveillance, which really involves ongoing assessment review. So typically you'll have experienced clinicians that will review the ratings that have been done. They'll review the administrations, and this can help to identify any issues that either an individual rater may be having, or sites overall, or maybe even issues with the protocol, in terms of the way things maybe have been explained or written. It'll help to identify those errors before they become too big and can really contribute to failure of the study. And in addition, by identifying these things early, that allows for retraining of the raters if necessary, allows for greater clarification of certain procedures, may lead to needed protocol requirements. So again, this kind of endpoint-specific monitoring is becoming increasingly common in trials. And here's just a few other points. A lot of times EDC systems will have automated checks that help the monitors kind of identify some of the issues. It's useful to have central review, as we often do, for imaging and biomarker data. Adjudication processes, again, where the clinicians kind of can get together, discuss ambiguous scores and how to handle them, how to handle missing data, for example. So again, just a few more examples of how this ongoing monitoring can really be helpful. And the last point I wanted to mention really is related to reducing endpoint-related burden for patients. We commonly will see trials that have a lot of assessments, not just scales, but a lot of assessments overall that are included, and it makes sense. We want to collect as much information as we can, as we think is useful. But sometimes it can be overdone a little bit. It can be kind of demanding for patients, and particularly if you think about elderly or cognitively disoriented patients, patients with other kind of disability. It can be hard to sit through long periods of assessments. And what you end up with is either patients that are not doing the best that they can, or not able to answer correctly simply because they're too tired. So you're getting noise, you're not getting an accurate representation of the patient. And it can lead to a lot of withdrawal, of course, which then again could really have a deleterious impact on your results. So there are a few steps that can be taken to try and mitigate this. One is the ordering of your assessments to try and minimize fatigue. So, for example, if you've got complicated cognitive assessments, you typically want to have those first, if you can, before maybe other subjective symptom ratings. Because you want to get the patient at their freshest, essentially, to complete the cognitive scale. You might try decentralized endpoints, wearables and things like that can collect data at home or with less interaction. Regular reminders and check-ins with patients. Selecting endpoints that are important to patients. I mean, sometimes we have a tendency to overlook that. Maybe in a particular disorder, we're wanting to look at how the drug impacts cognition, but what's really important to the patient, or maybe the patient's caregiver, is their behavior. Maybe the patient's agitated or aggressive a lot, and for the caregiver, that's really very important in terms of management. So, being able to get that input from patient groups, advocacy groups, to know what's important can really help with patient and caregiver buy-in and being able to accurately answer the questions and the endpoints. And then finally, just thinking about what endpoints are necessary. Thinking about, okay, what do we actually have to have included here to collect the data that we need, and what can we maybe leave out just to make things again a little bit easier on the patient. So I think we've reached the end of the slides. I think we do have a couple more minutes for questions. So thank you everyone for listening, and I'll turn it back over to Heather.
Okay. Well, thank you so much. I will pop back on, and thank you so much for that wonderful presentation from both of our presenters. We are going to get to a few audience questions. We have a little bit of time. If we do not get to your question, no worries. We will send these on to our presenters and they can answer them post-event. But I will bring back our presenters, and let's get to our first audience question. And this question is asking, what are your considerations on digital endpoints and wearables? Who can help with this one?
I think I can. Yeah. It's for either one of us, Marty. I can.
You want to do it? Okay, go ahead.
Yeah, I can add to it. I mean, so digital endpoints, they usually have the advantage of providing a very objective data with less rater bias. But so that's the plus, right? But they're only useful if they actually measure something that it's mechanistically related to your drug, and they're validated in your indication. Then you also must have a plan for dealing with the missing data and the patient compliance, depending on what kind of wearables, could be difficult for the patient. It depends on what populations. Some population can tolerate better some wearables than others. So if you fit this bill, if you fit these conditions, then it's definitely a good possibility. And Marty, feel free to add.
Yeah, I was just going to say, certainly, digital endpoints, I think, are in the earlier stages of validation, but they can be useful, especially for certain conditions. You think about movement-related disorders, being able to capture things like gait, for example, or tremor on an ongoing basis, or sleep, can be very, very helpful. So there's more research that needs to be done, again, to really validate them. But I think they're increasingly becoming an important part of the endpoint battery.
Okay, well, thank you so much. And unfortunately, that is our time. We've gotten to the end of the hour, but a lot of great questions. And again, we will forward these on to our presenters to answer off this event. So thank you so much to both presenters for sharing your time and your knowledge. And thank you to the audience for joining us today. This will be available on demand. You'll receive a link via email as soon as it's available. Take care and have a wonderful day. See you next time on Clinical Leader. Goodbye.
Thank you, everyone.
Thanks, everybody.